Thanks. I hope people file back in. I hope you had a good break. Before we turn things over for session 3, actually, as we turn things over to session 3, I want to introduce Dr. Holly Lisenby, who you already met, but she is the director of the Division of Translational Research at NIMH, and I'm going to let her lead off session 3. So Holly, take it away.
Thank you so much. I'm going to start with a brief recap to set the stage for session 3. And to me, the tagline of the day so far came from Misaki, who said, "Neurofeedback can translate neuroscience knowledge into brain intervention. Neurofeedback can evolve as neuroscience evolves." In session 1, we learned from Stephen LeConte that the heart of fMRI neurofeedback is the MRI, both its strengths and its limitations. And we learned that fMRI neurofeedback has a lot of knobs. From Masaya Misaki, we learned that because there are a lot of knobs, a better understanding of the mechanisms by which this form of feedback works could inform how we set those knobs. We learned that the mechanisms involve learning, which engages distributed networks in the brain beyond the region of interest that might be being targeted. And that with real-time fMRI feedback, we get 2 for 1. We get both the neurofeedback signal, but we also get the whole brain activations during the training, which may be useful in understanding differences in clinical outcomes. We learned from Michelle Hampson that real-time fMRI neurofeedback has the potential to shift long-term trajectories. This may be especially important for youth because it can represent a safe, medication-free way of leveraging the plasticity in young brains that could lead to a lifetime of benefit. But we also heard from her important challenges, like the need to support early-stage studies to identify the best interventions, and she argued for a reduced emphasis on statistical power. In session 2, we saw some examples from Susan Whitfield-Gabrielli about the promise that real-time fMRI feedback could bring for neuroprevention, neuroprediction, neuromodulation, neurotriggering, and network therapeutics. We learned from her that it takes a village. We saw the products of multidisciplinary teams and decades worth of work. And we understood the potential of combination interventions that combine the real-time fMRI neurofeedback with other forms of intervention like mindfulness meditation. From Karina Kovuto, we learned about how to hit a moving target. And we saw really exciting data that real-time neurofeedback intervention could decrease suicidal ideation in youth at risk for suicide. And then from Kim Young, we learned about challenges in this field having to do with dose determination and clinical implementation. We also heard a discussion about where does real-time neurofeedback belong between the psychosocial intervention space and the device space. We heard about pros and cons of each. And we heard about the importance of ensuring that neurofeedback work is optimally reviewed by those with the proper expertise and optimally supported on its pathway to clinical implementation, especially given its exciting promise. Now in session 3, we're about to hear from experts at the FDA from the Center for Devices and Radiological Health to address our questions about what role FDA plays in getting neurofeedback technologies from the stage of research into the clinic. And we're also going to hear from a team that has successfully navigated that pathway to bring a neurofeedback technology to the market. Bottom line up front, the FDA provides a pathway to allow devices like neurofeedback to get to the clinic by being shown safe and effective for an indication for use in the treatment of a mental illness. FDA does not control reimbursement, but it is a step in the process of getting treatments into the clinic. If you want to market a neurofeedback technology for the treatment of an illness, you do need FDA approval. If you want to use it off-label, FDA does not regulate the practice of medicine, but you will not be able to market or advertise that you are offering that technology. Insurers might decide not to cover the intervention in the absence of FDA clearance. But even with FDA clearance, they still might make their own determination to cover it or not to cover it. Now I'd like to hand the session back over to Chris Sampote, who will introduce our speakers, and I'll be back to moderate our discussion.
Thank you, Holly. Appreciate your comments and the recap, and also thank you for joining us today and for supporting this meeting today. We have three presenters and a moderator and a discussant today for session 3, starting with Dr. Talma Hendler, who's from Tel Aviv University. Dr. Hendler is also associated with Gray Matters Health, which represents a company in which she leveraged her research to develop an intervention to be brought to market and impact mental health outcomes. And she'll be also talking about that work. Also, Dr. Doi Kumsa and Dr. Nita Bajaj from the Food and Drug Administration will talk about the regulatory pathway that exists for interventions like real-time fMRI neurofeedback. And then finally, Dr. Luke Stokel from the National Institute of Aging will be joining the discussion that will be led by Dr. Lisenby. So with that, Doi, are you up next? Do I have you leading us off?
In the schedule, I'm going second, but I can go if that's preferred.
I think I'm the first one, Chris.
Very good. Talma, why don't we have you share your slides and you can lead us off and into the afternoon.
Thank you. Can you hear me? I hope so.
Yes, yes.
Okay. So my title is Crossing the Bridge from Idea to Biomarker, but I could also say that it's crossing the bridge from science to market, and this was thanks to a spin-off company that established for my work, and I will talk about the path that I took to get to this point. So this has been already mentioned. We are facing a great need, unmet need in mental health, and you have here the numbers, really incredible numbers, that patients that are looking for answers and healing options. Under these gray bubbles, actually, we have quite a lot of science that has been accumulated in the last 2 decades in animals and humans, of course. And with respect to the RDoC idea, we could color this unmet needs with some notions, some scientific notions that we have about what is the underlying mechanisms of these different disorders. And I mentioned here 3, but we could think of others. Motivation behavior, which is from the positive domain, positive balance domain, the cognitive control system, which is the cognitive domain, and the emotional regulation system, which is the negative domain. And I will focus in my talk on the blue one, the negative balance domain, which is related to anxiety, PTSD, and stress resilience. So in my talk, I will take you through the road of, I need to move things here. I will take you through the path I took from clinical need to lab work, and then from lab work to clinical application. So starting from scientific idea to biomarker. So that's the beginning of this is in a prospective study I did on the first responders who were expected to undergo traumatic experiences. And we found that amygdala hyperactivity was predictive of how many symptoms and disturbances they developed after the exposure to a traumatic experience. And this was following us replicated. So this is by now an established idea that the amygdala is important for emotional regulation, and it's probably related to the fact that it's highly connected subcortically and upstream to prefrontal cortexes. And in addition to that, what we know is that from accumulating studies that amygdala appears again and again in very different disorders, and gradually it has been acknowledged that it could be regarded as a transdiagnostic neural marker in psychiatry. And that was a good starting point in my mind to put forth the assumption that if we if indeed disturbances in the amygdala are related to psychopathology, we should try to modulate its activity. And since I saw the hyperactivity as a marker for trauma, the first direction I took was to try to downregulate the amygdala and hoping to see changes related possibly to emotion regulation. So clearly the best way to measure amygdala, as we all know, is functional MRI, and we heard a lot about it. But if we want to move to scalable intervention, and particularly I was asked by the funding agent that helped me do this initial work to actually demonstrate that I can do the work in the field with the first responders. I was thinking maybe I should move to EEG, but EEG, as far as I knew at the time, has poor precision and is not able to actually probe the amygdala. And that led to the idea that we should combine fMRI and EEG and try with machine learning to predict the fMRI from the EEG. And we did that about more than a decade ago, demonstrating that we can actually obtain electrical fingerprint from the fMRI that can be used as a probe in neurofeedback. And you can see here the very first results we got, test versus FEM, that people were actually able to reach this fingerprint mentally and downregulate it. And then, of course, we had to validate and prove it. And we invested a lot of work for almost 2 decades with many papers that you can just see some examples here, showing that if we bring new group to the lab and ask people to downregulate the fingerprint in the fMRI, we actually get back indications for modulation of the probe in the fMRI. And we could also demonstrate that it's probing a relevant process like emotion regulation using Stroop tasks. So of course, I don't have time to go into the details, but importantly, there is a lot of science behind that. Another direction that we took in the development in the lab was related to developing interfaces for the feedback that could be good activators of the amygdala. It was actually mentioned by Kim, and I was excited to hear that activation of the target is actually very important. And we were able to we developed an interface which is noisy interface. Maybe you cannot hear. I'm not sure you heard the sounds. Did you hear it? Because I didn't tick the note. Could you hear the sound?
Yes, we heard it. Yes.
Okay. So I'll just show it for a minute again. So this interface is continuously changing with the brain. I will stop it for the sake of time. And the more people downregulate the amygdala, the quieter the room become and the avatars sit down. So altogether, this brought us to the point that we can, and of course we demonstrated that people can use this to train the brain to downregulate the amygdala. And so I just kind of briefly demonstrate to you how we went from a scientific idea all the way to biomarker, and on the way also improve the technology of the biomarker, but also of the therapy. And now I will talk a little bit on the second point of going from the lab work to the clinical application and actually demonstrating it's actually has an efficacy in the clinical work. So the first indication that we looked at was trying to see whether we can enhance resilience among first responders, which were our first first target in developing the technology. And the idea was, as already I said, that if hyperactive amygdala is resulted in more post-trauma symptoms, we should try to downregulate it. And we undertake a very large study in the combat soldiers, and were able to demonstrate that indeed the soldiers in 6 sessions learn to downregulate the electrical fingerprint better than if we used another control condition. And most importantly, this was associated with improved alexithymia. Alexithymia is a marker of emotional regulation, of people knowing what they feel and how they express their feeling or understand others' feelings. And it has been associated with stress and trauma. So downregulation of the amygdala biomarker resulted in both learning and change in process. We also, so I could say that we target a mental process that was relevant to stress, but we also were able to show, bringing the individuals to the lab and ask them to now do real-time fMRI on their amygdala. We were able to show that they are actually better in downregulating the amygdala, but when they do it, they're also getting better in connecting to the ventromedial prefrontal cortex. And this has been shown by others that actually downregulating the amygdala is related also to improving connectivity with VMPFC, a very important node of regulation, of emotional regulation. So we talked about what is the outcome and the target. So here are 2 examples. And the next things we did is to try to prove that it's actually valuable for patients. And we performed 2 randomized controlled trials on PTSD, on chronic and complex PTSD, altogether 93 patients. And we were able to show, looking at the CAPS, which is a common scale in PTSD for severity of symptoms, we're able to show, as you can see in the darker bars on the left and in the red bars on the right, that people actually getting better in improving in their symptoms in a significant way compared to no treatment group control. And when looking at PCL, which is a self-administered scale, we were also able to show what has been already mentioned here, very importantly, that after 6 months, people even get better. So if immediately we had 36% improvement in PCL in the test group, after 6 months, we have 55% of people getting actually in a more than 10 points change, which is significant clinically. So this was all the science we did before the company was established. So again, a lot of lab work and a lot of publications. And then we established the company, and the company, the first thing they did is trying to replicate this clinical study, which has just been published also. And you can see here the result that they received from the CAPS. So 67% of the patients overall showed significant improvement. And in some subpopulation, even 80% that people were under other treatments like SSRI. And 32% were actually in a remission rate with more than 50% in some population. And very importantly, that also has been mentioned before, more than 90% patient compliance and only mild transient side effects. So this was the study that we actually approached FDA for regulation approval. So the point I want to make is that we need, this is the FDA, and like has been discussed here before, this is a device approval based on a 510K, a predicate device. And so it's in the limitation of this predicate device, and maybe this will be discussed further with the FDA people. But now we have a device, PRISM for PTSD, that is available commercially in the USA and also in Israel. So to sum this part, from a clinical need to a lab work, and from a lab work to a therapy and FDA regulation. It's a long road, a lot of hurdles on the way, but it's possible. This is what I want to say. But at the same time, I would like to, and this was possible because of Gray Matters Health, the company. I would like to also say that it's not the end of what we have to do. There is a lot of improvement opportunities here. And I just mentioned it here briefly. First, specificity. We need to enhance target precision and disorder-tailored interfaces, like I already mentioned a little bit, for instance, trauma content or obsession cues. We need to improve suitability, which is identify good or poor modulators, optimize learning, and combined methods, like already mentioned. We need to improve scalability. Right now, the treatment is given in the clinic. Maybe we can go home carefully and systematically study how to do it the best way and still be effective. And then we can improve setting about preparation, engagement, integration. I will just mention briefly these 2 points about the future, how we can go farther beyond where we are now. And this can be done, should be done in the lab, but the company can also help fasting some of the processes of this development. So more targets and claims. Like I said, we started with PTSD, but we are thinking of other claims, of course. And these bubbles need to be mapped completely. We need to have actually a library of biomarkers that can be used to probe different disorders and the combinations of them, of course, as well. We need to improve precision of biomarkers, and this has been already mentioned as well. Try to develop biomarkers that are process-specific, more and more process-specific, and use networks, not only regions. And this has been mentioned very nicely in the first and the second sessions. So we should definitely go in this direction and definitely try to also scale it to EEG, and it's possible. And the other thing is enhanced process probing by the interface. This is more about suitability, becoming more and more personalized about what exactly we activate when we are doing the neurofeedback. We already heard that this is important for the outcome. So again, we can use different interfaces that can probe different processes, like phobic condition or social anxiety or addiction. Lastly, we should increase personalization by accessibility, but also by understanding who is the good modulator. And this is just very briefly showing you here a study. And also to wrap, I wanted to finish and say that although we are now having a biomarkers, an EEG-based biomarker, we still think fMRI studies are incredibly important. And we are trying to combine the two worlds in order to understand the mechanism with the aim of improving and optimizing the learning. So if you are doing many sessions now with the electrical fingerprint, with a biomarker, you can perform fMRI before and after and learn about the mechanism that is related to the change in the EFP. And you can see here that people are actually, with time, this is EFP learning, with time becoming better. So this is a capacity of neuromodulation. And using a large, combining a lot of data that we had in the lab with people undergoing between 6 to 12 sessions of EFP, down-regulating of the amygdala, and also having fMRI neurofeedback at the end, we characterize a network. This was also mentioned by Messiah Stock. We characterize a network that is important for the modulation, and we demonstrated that this network is better predicting than just the amygdala alone the outcome of the EFP. So there is the relationship between these two worlds that we can actually learn from one world to another and improve the EFP with the fMRI. And lastly, not less important, this also predicted, this network the amount of change in alexithymia among these patients. So it's also probing into the process, not only into the modulation. And this is capacity, how much you can modulate, but we can also think about, we know that neurofeedback is a learning mechanism. There is a lot to study there and using models in order to improve it. And thank you. I would like to, thank a lot of my group, people from my group and collaborators and funders. Thank you.
Thank you, Dr. Hendler. Let me now introduce Dr. Doi Kumsa and Dr. Anita Bajaj from the Food and Drug Administration. Doi, please feel free to upload your slides and take us into the next session.
Okay, sounds good. 1 second. Am I sharing the right screen?
Yes.
Okay. All right. Okay. Thank you for having us. I'm Doi Kumsa. I'm a lead reviewer here at the FDA. My colleague, Dr. Anita Bajaj, and I will be presenting regulatory perspectives. And so we'll start with, and we have no financial disclosures to report. Trying to advance the slides. Okay. We are located in CDR-rich. Sorry, 1 second. I'm having some technical issues. All right. We're located in CDR-rich, the Center for Devices and Radiological Health. It is one of the 7 centers at the FDA, and within CDR-rich, which has super offices depicted here. We are in the Office of Product Evaluation and Quality, which itself is then, the super office is then divided into several offices shown here, and we are in OHD 5, Office of Neurological and Physical Medicine Devices. So this slide is just to give you an overview of the structure of our particular office. The Office of Neurological and Physical Medicine Devices. It's depicting the 2 divisions within it and the multiple teams under the 2 divisions. There is a lot of information, but we wanted to highlight that depending on your device and indication for use, you will likely submit to one or more of the teams here, and the assistant directors for each of the teams are shown below. We belong in the red highlighted one, the neuromodulation psychiatry team. We look at, when they have psychiatric indications, deep brain stimulators, vagal nerve stimulators, computerized behavioral therapy devices, whenever all of them have a psychiatric indication. So moving on to the regulatory landscape. So here we are looking at different classifications. We have class 1 to class 3. The class a device is in determines the extent of regulatory control. There are 3 categories. They were established through the FDA, the Food, Drug, and Cosmetic Act. So examples of devices in each category would be for class 1, they could be bandages, medical gloves, surgical masks. For class 2, there could be transcranial magnetic stimulation devices, transcutaneous electric nerve stimulators, biofeedback devices such as neurofeedback devices. Class 3 is there are high-risk devices or devices where we could not come up with what we call special controls. So when I say high-risk devices, they're implantable, such as deep brain stimulators, implanted pacemakers and the like. There are regulatory controls in place for all devices, and they're needed to provide reasonable assurance of their safety and effectiveness. The general controls, they apply for all devices, and they're needed to provide reasonable assurance of safety and effectiveness. But for class 2 devices, the general controls by themselves are not enough. So in addition to the general controls, they're also subjected to what we call special controls. This is because if they just satisfy, electronic establishment, registration, device listing, that's not enough to provide reasonable assurance of safety and effectiveness of the device out in the market. And special controls are usually device specific and indication specific, and they could include performance standards, post-market surveillance, patient registries, special labeling requirements. They would be listed out in the performance standard. There could be pre-market data requirements, and there could be special guidances with respect to that type of device. For class 3, which are the highest risk, for those, both general controls and performance standards by themselves are not enough. So in addition to those, they have to have significant clinical performance data. Some devices, while their risk level might not be high, if we are not able to come up with special controls, they could be in class 3. So that is kind of like an interesting situation. Okay, so we recommend that you start interacting with us early on prior to starting your clinical study, so that we can give you feedback on your clinical study design approach. You're able to do this through Q-subs. There are different types of Q-subs. A pre-submission, for instance, is one where you would submit your questions to us and you could get written feedback from us. It does provide an opportunity for you to interact with us early on and discuss with us your intended pre-market submission. So we don't do a pre-review of data in a Q submission, but rather it'll be feedback on your approach. So you could be asking us about a clinical study that you're designing, or it can just be an informational meeting too, where you share information with the FDA without expectation of feedback. Those are called informational Q-subs. And after that, typically, it's ideal that you do this before you submit an IDE or a marketing submission, so that we can provide you feedback, so that you can come up with a well-prepared submission. But sometimes it doesn't. So after this, you either submit an investigational device exemption or a marketing submission. So an investigational device exemption, and IDE for short, it's different from a marketing submission. So it pertains to devices that have not been approved. An IDE pertains to devices that have not been approved or cleared for marketing, or devices that are being tested for a new indication. So they're cleared and marketed for something else, but you're investigating them for another indication for use. And if the device or study is considered to be significant risk, then an IDE is needed. So when is a study considered significant risk? At the onset, the sponsor investigator, and the IRB are responsible to make a risk determination. And they are supposed to look at, based on criteria that are specified in a regulation. So basically the criteria are, one of the criteria is it intended as an implant and represents a potential for risk to health or safety or welfare of the subject? If that's a yes, then it's a significant risk. If it's sustaining human life, then it's a significant risk device. If it's for use of substantial importance in diagnosing, curing, mitigating, or treating disease, then it's a significant risk device. And then there's a catch-all caveat, which says otherwise presents a potential for serious risk to health, safety, or welfare of a subject, then it becomes a significant risk. So in fulfilling the requirements set by the regulation, we do take into consideration not only the device, but also the target population the device is intended to be used in, and the overall study design. So, if the target population includes patients who are suicidal, even with risk mitigations put in place in the study design, it could be a significant risk. So it depends. There are different types of IDEs, early feasibility studies, which are on the early side. The device design might not be final. They could inform device design. It could be in a small number of patients. Then we have traditional feasibility studies, where the device, it's further along than the early feasibility and it's near final, but the focus is still on the feasibility and safety of the device. The pivotal IDE studies are where we are looking for safety information and effectiveness information. And then the other pathway is a marketing submission. So you could come in with a 510K, a de novo, a PMA, or an HD. I will go into each of these as to what they require. So 510K is substantial equivalence. A de novo is when there's no precedent for when there's no precedent, and then PMA and HD is for class 3 devices, but I will go into each one of these a little bit later. Okay, so so we are showing you here a regulation that might be directly applicable to neurofeedback devices. So this regulation has been around for a while. There are devices under it that have been cleared in the 90s and 80s even. So note that you can have a device within this regulation, but you're not specifically stimulating with electricity or magnetic fields. So the identification of the device, I think it states that biofeedback device is an instrument that provides a visual or auditory signal corresponding to the status of one or more of a patient's physiological parameters, so that the patient can control voluntarily these types of physiological parameters. And it does give you an example of what those are. So we thought it was important to put in that this takeaway that most mental health applications will likely not be exempt from a 510K, and they will likely fall in this regulation because one, there are precedents, and two, it does exist. The regulation does exist. They are devices according to our device regulation definition. So we wanted to kind of highlight that there isn't a confusion as to whether they're a device or not. And then there's also not a confusion about whether there are precedents that have been cleared under this regulation on the market. So I will hand off the presentation to my colleague, Dr. Anita Bajaj, who will present clinical study design considerations.
Thank you, Dilly. I wanted to start by going over some review points to consider in clinical trial design, and just 2 caveats. They're not intended to be guidance, and we would really appreciate if you reached out to us via the pre-submission process that Dilly touched on for specific questions about your regulatory approach. Next slide. So some clinical review points to consider. First of all, the need for a well-controlled study. It's important to think about what is the appropriate type of control that you want to use in your study to minimize bias, which could be associated with the placebo effect. Ideally, you want to consider a sham-controlled study. I'll talk more about that on the next slide. Another aspect that's important to consider is randomization, as well as blinding. Because of the high placebo effect that I mentioned before, observed in many psychiatric disorders, it's important to consider blinding of not just patients, but also investigators and study staff associated with a clinical trial. Having a SAP or statistical analysis plan is also important. It should include a formal statistical hypothesis and pre-specified success criteria. Those are key elements to help support study implementation and success. Finally, determination of outcome measures is very important. We like sponsors to consider validated disease-specific measures. It's important to pre-specify the amount of change between arms needed to indicate success. We value objectivity of outcomes and functionality. For this reason, we prefer clinician-reported outcomes for scales based on objective measures. It's important to also define the time frame for evaluating the primary endpoint. And we, again, strongly encourage Q submissions to review the clinical protocols that are intended for marketing submissions before initiating the study. Next slide, please, Joey. These are some of the factors that the FDA considers when evaluating sham devices. We like to see a sham which maintains blinding such that the patients in both conditions are unable to tell if they're in the active or the control condition. Also, promotion of subject retention. This is really important because participants in the assigned control condition often drop out of the study if they know that they are in the control condition. And lastly, matching time on task with the treatment application, as well as level of engagement. Next slide, please. Some more viewpoints. In defining the target population, we want to consider the type of psychiatric therapy delivered. For instance, what diagnoses are used as targets. So the type of psychiatric therapy delivered via neurofeedback might be, for example, major depressive disorder, obsessive compulsive disorder, substance use disorders, post-traumatic stress disorders. We've already heard these discussed today in previous presentations. Even among these disorders, there's also different levels of severity, from anywhere from mild, moderate, severe, also treatment resistant. It's important to be as specific as possible. Also important to note if the device is to be used adjunctively or with treatment as usual options, or if it'll be used as a standalone treatment, not for use with concurrent medications or therapies. Some of these considerations will go into how the study is designed to support the indication for use and the target patient population. If we look at the issue of adjunctive versus standalone use, when a product is meant to be adjunctive or an aid to usual care, the patients should be on treatment as usual options. The protocol should state what the usual care constitutes. And if patients are allowed to be on medications, the protocol should include a plan for how medications will be managed and monitored to lessen the impact on, to take it into account the impact on the patient's condition so that it doesn't confound the results. For a standalone device, the protocol should have specific plan for patient safety if the patients are not going to be on medication or other usual care options. They should also have a plan for how patients will be washed out of the medications or usual care that they are receiving. And it's important to incorporate into the study design ways to show that the device is effective compared to usual care. Next slide, please. For review of marketing submissions, we use a risk-benefit analysis where we consider other available treatment options for this target population, the potential of risk due to concomitant use of other medications or the interruption of medication usage, a risk analysis, a benefit assessment of the proposed intervention, and an assessment of uncertainty also needs to be factored in. The target population is also important to indicate. For instance, in a pediatric population, it's important to look at the definitions that CDRH uses. For neonates, from birth through the first 28 days of life, infants 29 days to 2 years, children are 2 years to less than 12, and adolescents are aged 12 all the way through the 22nd birthday. That's a little different from some others. So a study that will use persons from ages 18 to before the 22nd birthday is actually a pediatric indication as well. Treatment-resistant population is also important to consider, and it's useful to be specific about which other therapies have failed, and it's important to specify the DSM-5 TR diagnosis when applicable. It's also important to consider how the data obtained in the study will be leveraged. Is this for a pivotal trial, a marketing submission, for example, and whether a new brain target is being studied, and if so, we need data on this novel brain location. Next slide. When looking at how to evaluate safety and effectiveness in the target population, we consider the screening criteria. This means the inclusion and exclusion criteria. and they should reflect the target population mentioned in the IFU. If you are targeting patients who are already on some type of treatment, are you targeting a treatment-resistant population? Are you specifying the DSM-5 diagnosis and how it was diagnosed when applicable? It's essential to consider how to design your study to support the indication for use and the target patient population. Next slide. Study outcome measures from a safety perspective should take into consideration specification of safety parameters, classification of adverse events, how adverse events are adjudicated, how they're quantified, guidelines for reporting of adverse events, and long-term plans for study participants. Study outcome measures from an effectiveness perspective need to take into account predetermined effectiveness measures with the rationale. Alignment with the indications for use, again, very important. Timeframes for evaluating primary endpoints, such as targeting acute versus chronic conditions. Explicit hypotheses in studies with statistical analysis plans, and the use of validated, generally accepted measures. A robust protocol to monitor the study is also important. Usage of assessment schedules, clear informed consent forms with study timeframes built in, case report forms with adequate detail, and the use of DSMBs or SMCs, which are data and safety monitoring boards and study monitoring committees. Safety monitoring boundaries, also known as stopping rules, are also important, and sample size is important, whether we are looking at safety outcomes or effectiveness outcomes. Next slide, please. Again, outcome measures are critical. We must pre-specify the study outcomes, define the time frame for evaluating the primary outcome measure, and we recommend the use of validated and objective outcome measures. Establishing clinically significant study outcomes is an important consideration to support study success. Ways to look at differences between groups and outcome measures could include a comparison of means or a comparison of response rates, in which you need to define the responder and the remitter. Always keep the placebo effect in mind, as this could lead to bias in results. Keep also in mind that scales used in clinical practice might not be ideal for data generation in a regulatory setting. For example, in major depressive disorder, we might like to see the use of a clinician-reported outcome scale, such as the HAM-D or the MADRS. Next slide. I think I'm back to you, Joey.
Oh, okay. Yes. Okay. Thank you, Anita. So hand in hand with the clinical study design considerations that Anita had just mentioned, we also look at non-clinical testing considerations, and it's important to consider that they will be device specific. So what is listed here might not apply to, like if it's a digital device, there isn't going to be an electrode or sterility. But in general, these are the various elements that we take into consideration when applicable. If, for instance, a device is something that's already on the market, but it is being tested for a new indication, and you're coming in for an IDE or something, we request that you include a letter of reference from the manufacturer, from the device you're using, so that we can leverage that information. So moving on to different types of marketing submissions. I had touched on them briefly earlier. So we have what are known as, for instance, a de novo petition, which are usually, they end up usually being class 2 for a new device type with low to moderate risk. And when there isn't anything like it on the market, that is class 2 under 510K. So it's a new intended use. Or it's a different technology that raises different questions of safety and effectiveness, and the risk profile is also different. You end up needing to submit a de novo. Then we have our 510K pathway, a pre-market notification. So it's typically required for most class 2 devices. What they need to do is show that they are substantially equivalent to legally marketed devices. So this is different from a standalone evidentiary standard. So you are showing that you are just as safe and just as effective as the predicate device. If you end up extending outside of that intended use, then you're either a de novo, depending on the risk level, or you might move into a class 3 device. Okay, and then continuing with pre-market submissions, the other ones are pre-market approvals, which are for class 3 devices. It is based on determination standalone, device safety and effectiveness. So here you're not showing yours as safe and as effective as something else. You are providing standalone safety and effectiveness device, and it's a class 3 device. And then we have the humanitarian device exemption. It is limited in that it is for less than for 8,000 individuals. There are approval standards, safety information showing no unreasonable or significant risks, and then effectiveness is probable benefit, so it's slightly lower than the PMA. Okay, and then these are for any pre-market submissions that you are going to be submitting. These are the different elements that it would need to include as applicable, so that you need your indication for use statement stating what the device is going to do, what it's intending to treat. You have to describe the technology. You have to provide the device description, the different components, accessories. If you're adding something else to it as a component, that goes into the description of the technology. The labeling, of course, based on the outcome measures, on the outcome and the clinical study design that you have done, or any performance testing that you have done. It has to be reflected in the labeling. As applicable if it's sterilized or biocompatibility, if it's a software device or devices that utilize software or any kind of connection. We have regulations where software and cybersecurity documentation is needed. And cybersecurity documentation is becoming increasingly important, and depending on making sure that cyber devices that are able to connect are still safe. And then also you will include your performance testing, whether that be bench, animal, or clinical testing. This is just basically a summary of the different types of pre-market submissions. So if it's research and development ongoing, as we mentioned, we recommend you come in for a Q-Sub and ask about the pathways you're trying to pursue. You can submit a risk determination request as well. Your IRB might ask you to submit a risk determination request. You can ask for a classification, so whether you know which pathway you belong in, you can submit a 513G to get that. Investigational device exemption also belongs in this phase. It's in the research and development for ongoing. And then we have the cases where research and development is complete. So now you are entering the marketing arena. So depending on what the device is and its indication for use, it could be a pre-market notification, it could be a de novo PMA or humanitarian device exemption. And this slide is just showing that the review process is a collaborative effort. So we have, it consists of interdisciplinary teams. The team has several subject matter experts, including clinicians like Anita, different types of engineers, and different statisticians. We do have extensive interaction with sponsors to make sure the submission makes sense. We have, if there's anything that could be resolved interactively, we try to do that. We do interact with our policy staff to make sure the decisions we're making are aligned with existing regulations and policies and guidances. And then from time to time, we do have we interact like this meeting, external expertise, solicit external expertise, and there are like panel meetings as well. We have patient listening sessions as well. And this is our contact information. And we want to thank you for giving us your time, and you can take your questions.
Thank you so much. So I'd like to invite the rest of the panel members, Dr. Hendler and Dr. Luke Stokel from NIH to join. I'm going to start with a few comments, and then we're going to take a series of questions. The NIMH mission is to transform the understanding and treatment of mental illness through basic and clinical research, paving the way for prevention, recovery, and cure. Neurofeedback technologies, both EEG and real-time fMRI neurofeedback, represent an exciting avenue to contribute to this mission, which is why we convened this workshop. Starting with our end goal in mind, that of prevention, recovery, and cure, we are interested in supporting research that will accelerate the translation of promising interventions into clinical implementation, like neurofeedback. That's why we want all of us to have clarity about what are the steps that it will take to get neurofeedback technologies into the clinic. Since in order to use neurofeedback for the treatment of a mental illness, whether it's a DSM-defined diagnosis or a transdiagnostic symptom within a mental illness, FDA clearance is required to be able to market such a technology. So we want to make sure that the research that NIMH supports will provide the types of evidence that the FDA will need to see to make a clearance determination. Waiting until after a confirmatory efficacy study is completed before consulting with FDA about what level of evidence will be needed to clear a device risks hitting a roadblock that might have been avoided if one had consulted FDA earlier, ideally before designing a confirmatory efficacy trial. This is not to say that you need FDA clearance or even an investigational device exemption before doing a non-significant risk confirmatory efficacy trial, but rather consulting FDA when you're designing such a trial may accelerate ultimately obtaining the evidence that would be needed for a future 510(k) clearance. Now, we've got a lot of questions from our panelists and also from our audience online, and I'm going to start with a few, starting with a question to our FDA colleagues. You showed us regulation concerning biofeedback devices. Do I understand correctly that the FDA considers neurofeedback technologies like EEG neurofeedback and real-time fMRI neurofeedback as a device?
It depends on the indication for use. So if they're trying to treat or diagnose a disease or some psychiatric indication or any disease, honestly, yes, they would be a device.
So if you want to use neurofeedback for the indication of use for treating a symptom in a psychiatric population, that would require FDA approval. Am I understanding you correctly? I just want to be sure.
Yes. I because the question said, is it a device? So I was trying to answer both of them.
Okay, thank you.
Saying it is a device, and then.
Thank you. Next question. Am I correct that the types of EEG and fMRI neurofeedback studies that you saw presented in today's workshop likely would not require an investigational device exemption, assuming that the local institutional review boards had judged them to be non-significant risk?
So it is likely. So we do outsource that significant, non-significant risk determination to the IRB. So if the IRB determines that they are an SR, typically we don't come in and make it a SR. But if there is a difference in our judgment between the IRB and us, we are the final word in it. But typically it is outsourced to the IRBs. So when IRB determines an SR, we don't come and say it's SR.
Okay, great. This next question is for Dr. Hindler as well as the FDA. We saw an example of a neurofeedback technology that went all the way from the clinical need to the research to clinical implementation and marketing approval using an FDA 510(k) substantial equivalency mechanism. And Dr. Hindler emphasized the important role of the industry partner, Gray Matters, in making those steps possible. The question is, what stakeholders should be engaged regarding the next steps in the translation of real-time fMRI neurofeedback for therapeutic applications? For example, would MRI device manufacturers should they be involved? Would they be the ones to lead pursuit of a regulatory pathway? Anyone on the panel who would like to answer that? Maybe Dr. Hendler?
I'm not sure I'm the person to answer it, but I would say that having strong stakeholders in the game really helps to understand what is possible and what are the risks that are need to be taken into consideration when you try to commercialize something. This is issues that as scientists we have no idea about. I've been learned a lot in the way about this, and so I think if someone is thinking about commercializing a procedure in the fMRI, it will probably will be wise to advise with people who are dealing with these devices and have some experience and expertise and prior knowledge about what it takes to commercialize something like that, because it's about investment and risks, and we are not experts in that. So I would suggest to be very specific about what you are going. It's really, the question is regarded to what is your aim. Is your aim involve fMRI? Specifically, you probably need to talk with the stakeholders that have any idea about it.
Yes, Dr. Hendler is making a very good point. Marketing implies that you are a company that has commercialized a product that you want to sell, and you might be selling to health systems or providers who would then use that product in the treatment of whatever it has an indicated use for, according to what the FDA label says. And typically that's not the researchers, unless they, like yourself, are engaged with developing industry partnerships, startups that would then take that next step to commercialization. I see Luke, Dr. Stokel has a comment.
Yeah, I mean, it's not dissimilar. I'm also put this in chat for the other panelists, but you know, it relates to another related technology, which is prescription digital therapeutics, whether it's cognitive behavioral therapy or cognitive training elements in apps or in devices in hand, versus something like EEG that Dr. Hendler just presented. It's more a question about how the FDA thinks about approvals for those approaches differently. And I'm thinking, the reason I bring that up is to think, is there something about the device itself, whether the EEG technology, the fMRI technology, that is important for investigators that see themselves down the commercialization path where they want to bring something to market that would help them think differently about how they're designing their studies and conducting their research to get to market. I use the example of prescription digital therapeutics because to me, it's not dissimilar from the target mechanisms that we're interested in fMRI-based neurofeedback. So I guess that's maybe a question for FDA more than Dr. Hendler.
So I'm not sure which industry is well posed to kind of address it. I agree that it's very similar to digital therapeutics, like you're saying, but I'm not sure which industry is better, posed to address it.
Dr. Hendler.
Yeah, maybe just to add that it is also important to advise not only with the industries that are actually making the device and creating the platform, but also with the target population or the insurance company. I think Kim mentioned that as well. It's really important because that also affects the product and the marketing possibilities and will influence the stakeholders and kind of mapping the risk, I think. So this is in addition to when you are thinking about going ahead with commercialization.
Great, thank you. I'm going to move on to another question here from Dr. Kim Young. And Kim, if you want to put on your camera, you're welcome to, or I can just read this. Why now? Would you like to put voice to your question, and happy to address it.
Sure. Yeah. It just, for the past 20 years, we haven't been considered device-based, and we've been excluded from these definitions in literature reviews and meta-analyses. And so the question really comes to mind is why now? Why is it now that we're being considered devices when we haven't been previously? At least in terms of I understand the FDA defines us as devices, but NIMH has not included us as a device up until today.
So thank you for that question. And as you saw in the FDA presentation, there has not been a change in how the FDA views the technologies that deliver neurofeedback. Specifically, there's already regulation concerning biofeedback, and now an FDA 510(k) clearance for an EEG neurofeedback device. There has recently been an increased collaboration, I would say, between our agencies that's increased mutual understanding of how FDA views neurofeedback technologies. And we are attempting to be proactive, we in IMH is attempting to be proactive to use that increased understanding to accelerate the translation of the very promising neurofeedback research into something that would be viable in the clinic. Learning that this is a device-based intervention, learning that FDA approval is required for neurofeedback technologies to be marketed has been a very important point in shaping how we approach the intervention development pipeline for technologies that are viewed by FDA as devices. And Dr. Yang, you had a second question, if you'd like to pose that one. Or perhaps that was already answered. Yeah.
Yeah, I think it was just general confusion as to whether or not if we're for research studies for like R61s and R33s, if we need the FDA approval, or if this is really when we're ready to go to market, which I think has been clarified by now.
Yes, and just to re-articulate that, if the intervention is judged non-significant risk by the IRB, that would be not require an investigation device exemption. Dr. Kumsa did mention that if there is a situation where there's a disagreement between FDA and IRB about the determination, FDA is the final authority on that. But you did also hear her say of the studies that we saw presented today, it did not appear that those would require an investigational device exemption. And I'd like to also further underscore for all of the audience listening today, and Dr. Yang, you made this point very well in your presentation, the safety track record of real-time fMRI neurofeedback has been excellent. And so that's a very important point to underscore. Next question.
I do want to jump in. 1 second.
Sorry, go ahead, please.
For the studies that were shown, it does appear they're non-significant risk, but we don't know. We haven't seen them come in. So it's not an FDA recommendation or anything. They do appear non-significant risk. If the IRB called them non-significant risk, likely, but we don't know.
Great, thank you. And Dr. Yang, you did cleverly cite some of my own literature reviews on the topics of neuromodulation. They were not completely all-inclusive. So please don't interpret my failure to include neurofeedback as saying that it's not important or that it's not defined by the FDA as a device, because in fact it is. I saw, did Karina raise her hand? Please go ahead. And then Michelle Hampson.
I think that, first of all, I think that the road to FDA is unavoidable. I know that it's going to happen, particularly if we continue demonstrated that this has clinically significant results, which is what we hope for. I do want to caution that some of the populations that we work with are going to be naturally viewed by IRBs as very high risks for the institution that is hosting that particular research. And I'm not talking about just me, but many other researchers that really have in sight significant and salient mental health problems in our country and in other countries, like, increased suicide risks in kids and adults, which is very, very pronounced. But it also carries risks to the institution to run research in those populations, because we can't control, entirely the outcome of one or two participants, even if we do our best for it. So very easily the IRB will say, you know what, you are going to be a high risk study or a less than, more than minimum risk study. You need to bring your little fighting gloves and talk to the FDA. We leave it to you. And so the scientists are going to fly away from, I'm not going to touch suicide research. I'm not going to talk those things with anything. I'm not going to write any grants about that, because obviously it's not going to allow us to develop that area. I'm just giving a very simple example.
Yeah.
I hope it's helpful. So
let's use that example. So, and your work is very important when we think about the unmet public health need of preventing suicide, specifically in youth at risk. So non-significant risk determination, I'd like to ask our FDA colleagues here to comment. My understanding, and you can correct me if I'm wrong, is that the significant risk determination refers to the device when used in the specific study where it's going to be applied. And there are specific vulnerable populations, youth being one of them, that might be at higher risk for the side effects of a device when it's used in them. My understanding of that is typically, let's say, in my field of brain stimulation, if I was going to use a transcranial magnetic stimulation protocol that might put youth at higher risk of seizure, then because they're a protected population, that might require an IDE. But in this case, where the neurofeedback, it's not, the vulnerability of the population is not increasing the risk of a side effect from the neurofeedback. I don't think that.
No, but it's the perception.
I'd like to ask our FDA colleagues, because they can tell you more than perception. They can respond about the
Thank
regulatory.
You for willing to engage in discussion. This is productive.
I think, Anita, you're going to talk about the risk, right?
Sure, yeah, I'll start. We do like to look at the patient population, and of course, in a vulnerable population like persons with suicidality, we would be very cautious about the potential for that to be substantial risk. If the device itself does not hold the risk, but it's the patient population and their ability to be part of a study that could increase the risk, we do sometimes come to the determination of substantial risk. And it's just to add an extra layer of safety and protection for them, so that the FDA will be involved in the IDE process. I do know that it's a balancing act. Whenever you want to encourage innovation for devices that are serving these target populations that are very vulnerable, whether it's suicidal persons or, in another context, pregnant women, children, whoever, the need is high, but the need for protection is also high. And so the FDA will be very cautious about those populations when making the determination of substantial risk. Did you want to add something, Joey?
No, I think you covered it. I will put it in the chat. There's the regulation is 21 CFR 812.3M. It talks about when something is significant risk, and the last point kind of says otherwise presents a potential for serious risk to health, safety, or welfare of a subject. And it's kind of broad, so it's possible that we might consider it significant risk.
Oh, okay, thank you. So, and let me just say that, these regulations are meant to protect our human subjects. So let's not view them necessarily as barriers, but rather pathways to put the proper controls in place to protect safety, which of course we are all committed to. I want to go now to a question from the audience. This is, again, for our FDA colleagues, a question regarding 882.5050. Dr. Kumsa mentioned that most mental health indications for use do not fall under this exemption. However, current neurofeedback practice, which targets various mental health conditions, including depression and anxiety, is conducted under the prescription use part of 882.5050 exemption. Thus, neurofeedback devices are sold to clinics without a specific indication for use. However, the clinicians are able to use the device for various mental health conditions based on their expertise. So as long as the device does not make marketing claims that include specific conditions in its indication for use, then it can be sold under that exemption. Is that correct? In the case of PRISM, they made a specific claim for PTSD in their indication for use, which therefore required clearance. Is that correct?
Okay, so just to make sure I understand, if they're not making marketing claims with the indication for use, with this specific psychiatric indication for use, that it wouldn't, technically, you don't have clearance for it. So it is kind of.
So this was a very long question that I just read aloud to you. I just put it in the chat.
Okay.
So I think part of this is describing what's currently going on in practice. So you might buy a neurofeedback device.
Using something off-label, basically.
Off-label, okay.
And use it off-label. And right, the practice of medicine is not regulated by the FDA, so you can certainly use it off-label. But if you were to put it on your website and advertise, come get your neurofeedback for the treatment of XYZ disorder, that would not be consistent with FDA regulations. So am I correct about that?
Yes, you're absolutely correct.
So it's not to say you couldn't do it. We don't know if you get reimbursed for it. You know, it's as a clinician, it's on your medical license, how you choose to practice. You know, we're let me just speak about our mission at NIMH. We're interested in supporting the research that would lead to the development of treatments that are safe and effective and available clinically to everyone, widespread. So we're interested in supporting work that would ultimately lead to products that could be marketed, such that they would be available long-term for patients and reimbursed by insurance. Dr. Young showed some healthcare utilization numbers about how much MRI costs and 4 courses of CBT and so on. If a person had to pay for that out of pocket, then you'd be talking about a very small part of the population that could access that treatment. Dr. Hampson.
I just wanted to come back to the practical side of this a little for people who are doing like really early stage research, where they're not even like doing confirmatory efficacy trials, but are just really at an exploratory stage. It sounded like you were saying as long as it's minimal risk, you shouldn't really need to deal with FDA issues. But of course,
yes, that's right.
Okay, cool. But if you have vulnerable populations, or if you just have an institution that doesn't want to take any responsibility and they're just going to market as be super conservative and say, okay, call it moderate risk so that the FDA signs off before we sign off. What is to be expected, and what is like the most efficient route to try to get approval for something which is essentially pretty safe and very early stage? Like do we go to a Q-sub? I was a little confused with all the different terminology. But would you just go straight to applying for an IDE, or is Q-sub like a way you can just ask, does this even need an IDE or?
Yes, so like the best way would be a Q-sub, because we can help you put together a good submission. And then when it comes down, yes, a Q-sub would be the first step. If your IRB says you need to go to the FDA, then it means you need to submit an IDE. But prior to submitting the IDE, we recommend that you submit a Q-sub.
I'd also like to clarify, session 1 really focused a lot on how exciting these tools are as a basic science platform. At our session right here, session 3, is about using these tools to develop treatments that would be implemented in the clinic. So what we're discussing here right now does not impact the early stage or the basic science use of these tools. As long as you're using it in a context that would be non-significant risk, then you would not need an investigational device exemption. If you're further down the translational pipeline, where you're at the stage of a confirmatory efficacy trial and you're saying, we believe this study will lead to a new treatment for depression, that we want to have available for patients, that's when we need to start thinking, okay, well, how are we going to get that patient, how are we going to get that treatment to a patient? Is it a device? Is it going to need FDA 510K clearance? Will we ultimately need a company that would market this? So that's the kind of the transition from a research use of the technology into the clinical application of the technology. All of it's important, and I know it's confusing because we've covered a lot of territory from the basic all the way to the clinical implementation in today's discussions. Dr. Hampson.
Yeah, sorry, I'm still a little bit confused because what's not clear to me is, if you ever reach out to the FDA and say in a Q sub, does this even need to apply for an IDE? Do they ever say no? Like, no, you don't need to apply for an IDE.
I could tell you, I've done it myself, and yes.
You can submit a risk determination. So you can say, give me the risk determination for my study and device.
Wonderful, thank you.
And yes, I have another question here from the audience. Can we still submit a grant proposal for fMRI neurofeedback under a psychobehavioral approach? At what point does the NIMH plan to characterize fMRI neurofeedback as a device-based approach? Will it be based on the purpose of the study, specifically the stage of the study, such as early, middle, or late stage? For the NIMH's mechanistic clinical trials, they do not allow clinical outcomes to be included. What direction does the NIMH expect us to take as an early stage investigator? I'm a bit confused. Okay, well, thank you for that question. You've got a lot there in that question. Going back to, I guess, Dr. Hampson's question. So if you're using any technology to ask basic science types of questions, like what does the circuit do? Can we train people to modulate this circuit? If you're in this kind of the more basic question type of realm, no, you don't need to be generating the types of evidence that would support a 510K approval for a product. If you are trying to develop a treatment for a diagnosis or a set of symptoms within diagnoses, and you're trying to enter the clinical trial development pipeline the intervention development pipeline that we have at NIMH that supports early stage intervention development. So it's not early stage tool development, it's early stage treatment development. That's specifically the R61, R33 type of application. Then yes, we are aware that FDA considers using neurofeedback technologies for the purpose intended use of treating someone. They do consider that as a device, and we are currently at NIMH accepting such applications under our device-based funding opportunity, R6133 device-based. It's true that we have historically accepted them under the psychosocial intervention funding opportunity. We are currently accepting them under the device-based funding opportunity because of an increased awareness of how the FDA views such technologies, and because even at the early stage of developing these interventions, we want to be sure that we are funding the research that will generate the level of evidence that will be needed ultimately to get these technologies into the clinic. And we've learned today that FDA 510K approval is one of those steps. Yes, Dr. Hendler.
I would like to maybe make a point here about academic freedom and the exploration that has been discussed by Michelle so nicely, that as scientists, although it sounds good to say, oh, if I talk to the industry people beforehand, I could have done this and that and all, or maybe other things, but at the same time, it would have maybe kind of constrained my decisions to do something that is only safe and actually already see the target. Some of the success comes from just trying out sometimes even crazy idea. When I thought about combining the EEG and fMRI, everybody said, no, it's really not possible to see amygdala with EEG. And if I went at this point to an industry person, he might say, this doesn't sound reasonable. Maybe start with the motor cortex. So I give it as an example because it's easier to correlate it with the EEG, with the fMRI and EEG. So I took this very big jump without talking to the industry people, and that actually allowed me to kind of discover a new possibility. So it's a very delicate line between not going all the way crazy and doing things that are unrelated to the end point of a commercial product, but also letting researchers to try things that maybe not immediately look as they can be FDA approved, but if they prove to be valuable, they will. So that's my point.
Thank you.
Can I bring just a one small point? And this is a point that is, as Talma and other notes, kind of critical in that movement from, science to implementation, is the importance of replication. And I try to not very subtly put it into my talk because it is something that does plague our field just by the fact that we often reward positive p-values less than 0.05, but negative or absent results are not published or not known. And I was just going to take this opportunity to suggest to NIMH that you find a mechanism or a way, even if it's just putting posters or submitting a lost dissertation of a student that spent their entire career trying to find something and they didn't, the data gets lost. And it would really be helpful to have a site or a place for negative results because they're extremely important to guide what comes after. And I know that I'm preaching to the choir here, but I just want to make everybody notice how much our milieu prices first-time results that are significant, and how much non-significant findings are squelched, basically.
Yeah, you're making a very good point. And I want to parse it a little bit about negative result. There are negative results that are informative because you have disconfirmed a hypothesis. And then there are negative results that might be because of being underpowered. And those are two different types of negatives. But the general point is that there is a publication bias, and addressing that would be important for all of our aspects of science. I realize we're going to have time just for a few more questions, so I do want to get to a few questions from the audience before we hand it back over to Chris for the wrap up. This is a question for our FDA colleagues. You've been using the term disease. Can an FDA clearance be given for symptoms? How does the research domain criteria terminology fit into this?
I think it's sort of, it would be a clinical kind of different definition. So if clinically it's determined to be this, we've cleared devices for treating symptoms of a disease. So it's possible.
Okay, so you can, and in fact, my understanding of the PRISM approval is that it is for relaxation and stress in people with PTSD, not for curing all of PTSD. So that's an example of treating symptoms within a condition. Great. I have another question here. Since we use many free software programs like AFNI for fMRI neurofeedback, I am uncertain about how we can commercialize this approach. AFNI is intended for research use. And that's a nice segue to talk about the software part, because FDA has a whole classification for software as a medical device as well. And we saw applications of machine learning and artificial intelligence approaches to be able to do the signal extraction. Dr. Hendler?
Yeah, maybe I just it's a good point. And I was actually surprised that everything I did had to be redone in order to be submitted for regulatory. So everything that the company is now selling is not done in my lab. It was looking at what I did and doing it again and maybe improving it on the way. But so that's, I think, the answer. It needs to be redone in a documented manner, so it can be submitted as expected to the regulatory agencies.
That's the type of roadblock that we're hoping to avoid by knowing in advance what types of evidence would be needed or documentation for the studies that NIMH supports, so that the NIMH support can optimally facilitate and accelerate the ultimate marketing and commercialization. So thank you for that. I do want to take one more question. Can the FDA please clarify further their position on the need for controls? The PRISM for PTSD device was cleared using a single arm study, though there were pilot controlled studies. The Bridge auricular stimulation device was cleared based on a single arm study for opioid withdrawal. The Neurostar was recently cleared for adolescent depression based on real world data despite a failed multicenter trial. When are controlled studies needed and when are they not needed? This is very confusing. At one point I do want to make about the recent FDA clearance for the adjunctive treatment of depression in adolescents with TMS is that was an extension of a label of an already cleared device that was already cleared for adults. Label extensions are treated somewhat differently than an initial approval of a device. But would our FDA colleagues like to comment on this question?
I think Anita will chime in. So without going into specifics of typical sponsors or companies, we always try to, we want to look at performance data that shows us that we can attribute effects to the device. And the main way we can do that for most psychiatric devices is we need a control that would help us account for the placebo effect. And if we can attribute an effect to the device, that is kind of like what we base our determination on, that and the benefit risk analysis. So we are always recommending, as Anita had said, RCTs that are placebo controlled, but I'll let the clinician speak.
Yeah, I mean, I was just going to say the same thing, which is that the gold standard is the double-blind, sham-controlled, randomized controlled trial, but you know, each indication has different considerations and might have a different type of requirement based on the risk-benefit profile that we come up with for that device. So it's hard to give a blanket statement.
I'd like to thank our panelists from sessions 1, 2, and 3 for fielding these questions. And I'd like to hand it back over to Chris Sarampote for our wrap up and concluding remarks.
Thank you, Holly. And I'd like to add my thanks to every single one of the presenters who joined us today and who shared their research, shared their perspectives, shared their history, and their work to impact the lives of folks affected by mental illness. One of the things that struck me as we talked today was the range of issues that even if you take a narrow focused area, such as intervention development of neurofeedback or real-time neurofeedback, you see that the types of science that involved everything from basic to engineering, to clinical trials or to services and interventions and to regulatory implementation. It takes a wide range of expertise to develop an intervention and to make an impact. But not only that, it takes quite a bit of time. And there are people who have devoted their entire careers, and we thank you for sharing that, or people who are just starting their careers. And we had a number of questions in the chat from folks who are just starting out and wanting to know how to engage in this field. And so we know it takes a lot of time, not only a broad range, but also a lot of time to do so. So we thank you again for all your work and for sharing that. Couple quick things I just wanted to add on to that is at NIMH, we are very eager to hear from researchers who are proposing to conduct studies. And I would encourage you to contact program officers early and often to talk with us about your ideas and your aims. We're happy to give feedback. We find sometimes that people don't realize, oh, I could have contacted my program officer. We'd love to talk to you. It's one of the best parts of this job is talking to researchers like you. The second of all is also reach out to FDA. FDA is also there to help you with your products, to help you with your interventions, and help you think about how you might get approval and how this might ultimately be a benefit to folks. I love the discussion that folks have had here, and I know that this is a tight-knit community, and we probably no need to do this, but I would encourage you to keep working together and collaborating, particularly on the issues that you discussed today. And particularly as you think about this last question about how do we take issues related to regulatory aspects and think about them on how we design our studies early on, I think it's really important, an opportunity for collaboration. Just as so some folks, as I mentioned earlier, we will have a summary, transcript, and a recording posted in approximately 2 weeks on the NIMH website. And to that, I wanted to thank our contractors, Deborah and Susan, and also our transcript folks who have been working with us today and who have been working behind the scenes before this to set up the meeting today. Thank you all very much. And then lastly, I'd like to turn it over to Michelle. I know you wanted to share just a little more information, and just to thank you, Michelle, for co-chairing this. You were tremendous in helping us to think about neurofeedback and real-time fMRI neurofeedback and its role in the future of NIMH funding. So thank you.
Thank you, Chris, for all the awesome work you put into this. What a great organizational feat. And also to everyone, it was really helpful to have people from the FDA actually directly talking to us, which was a unique opportunity. I just wanted to share my slide for the meeting again, but I seem to not have sharing. Oh, great, just received it. I just wanted to put up a little reminder to everybody, or if they missed my talk, that the big meeting in our field is called RT-FIN, Real-Time Functional Imaging and Neurofeedback, and the next one will be held in November in Heidelberg, and we'd love to see people there. Thank you.
And I'd like to thank our co-chairs, Dr. Hampson and Serapote, for doing a wonderful job, along with all of our speakers, the logistical support, and all of you who attended today. To quote again, Dr. Masaka's comment, neurofeedback can translate neuroscience knowledge into brain intervention, and neurofeedback can evolve as neuroscience evolves. And as you've seen here today, regulatory agencies and funding agencies can also evolve as the science evolves, because we want to support your best ideas and move them forward towards improving the lives of people living with mental illness, their families, and communities. So thank you all.
Thanks, everybody.
Thank you.
Thank you. Bye.
Thank you.